triage technology can be used to rapidly screen the presence or absence of a specific target in a sample. This process will be done using the magnetic nanoparticles or MNPs developed by Dr. Evangeline Alosilha of the Nanobiosensors Laboratory at Michigan State University. So what are these magnetic nanoparticles or MNPs? One of the MNPs synthesized by Dr. Alosilha's lab is made from ferric oxide. As such, the ferric oxide nanoparticles confer the MNPs its characteristic supraparamagnetic properties, meaning the nanomaterial becomes magnetic in the presence of magnets or magnetic field while remains colloidal in its absence. The MNPs can become a powerful and useful biosensor for different kinds of targets. As such, to confer specificity during interaction with these desired targets, the surface of the MNPs may be functionalized with different biomolecules. This may include peptides, glycans, antibodies, or even oligonucleotides. In the case of the MNPs for this study, glycans are being used as the surface modification. These MNPs can be used for the detection of various target pathogens such as viruses, bacteria, or even fungi through a process known as matting. So how does matting work? A sample of the pathogen, and in this case a virus, is added into a 1.5 ml tube containing the MNPs. Then, the solution is mixed gently while avoiding the formation of bubbles. The tube is then placed into a magnetic rack. The mixture is incubated for 5 to 10 minutes at room temperature. During this period, the molecules on the surface of the virus interacts with the ligands of the MNPs, resulting to the aggregation of the MNPs and the viruses. Moreover, the MNPs are attracted by the magnet on the rack. Hence, a matting pattern is formed and observed inside the tube. The supernatant is carefully removed and the tube is gently lifted from the magnetic rack. Notice the difference between the aggregation of the MNPs in positive and negative samples. The matting pattern shown on the left is indicative of the presence of the virus in the samples, whereas the tube on the right tells us that the virus is absent. Pictures of the resulting matting patterns were taken using a smartphone camera and will be subjected to analysis using the ImageJ software. The matting area, width, and height will be measured to characterize the patterns. Moreover, the differences in these parameters can be possibly used to distinguish the different target pathogens. Therefore, MNPs can become a useful tool in detecting the presence or absence of specific targets, and it offers great economic advantage due to its low cost. We would like to acknowledge the following individuals who are behind this promising project. Dr. Maria Anita Bautista, our program leader. Dr. Mark Pierre Dimamay, our Project 2 leader. Dr. Divina Amalin, our Project 3 leader. Dr. Lilia Fernando, our study leader. Mr. Michael Sandino Flores, the research assistant, and of course, Dr. Evangeline Alosilha, our consultant. We are also grateful for the trust and support of our funding agency, the Department of Science and Technology, Philippine Council for Health Research and Development, or DUST-PCHRD. This project is currently being implemented by the De La Salle University Manila together with our collaborators.